Good morning, everyone. I hope that you spent some interesting time working on the first two lessons of Unit 2. As you have noticed, we are now working on the processes that take place in living organisms. So today's lesson on day 6 will be on transport across the cell membrane. These are the daily reminders that we know. You know that today we're going to have our Microsoft Teams meeting number 3, and this will happen at 11. Please remember to check the announcements as well as the activity feed. If there's anything that is worth sharing, I will share it there. And remember that yesterday I added a tracker sheet. So every day you should go to the same link and move your name into the lesson on which you're working to make it easier for other classmates to find people who are working on the same lesson. The due dates, we have discussed this during the last meeting, and as I said, the hard deadline is at the end of each unit, but the final date for everything is July the 28th, after which no assignments will be accepted. So if there's anything that is still late, submit it anytime up to the 28th. Of course, it will make it much easier for me while marking them to be just at the end of the unit time, but it's okay till the last date. So we are still in Unit 2, Biological Processes, and we'll start with transport across the cell membrane inside and outside, and then photosynthesis. This is our agenda of the week, and this is our work at a glance. So as you can see, I tried to have less must-dos than before. And for the should-dos, just read them, go through them, but you don't have to submit the assignment. But just remember the knowledge and the prior knowledge, the different skills that you might be needing in your must-dos. They're discussed in the should-dos as well. So if we go now to our topic. So I would start by checking the words and see what I remember, what I can put together. And then remember that transport across the membrane occurs from inside to outside and from outside to inside. So the membrane, the cell membrane, is the active structure through which transport is taking place. So understanding and remembering the structure of the cell membrane, I'm sure that you have already studied this in grade 11, but this is a good reminder of the phospholipid bilayer and the proteins that shield the polar substances from the tails and allow them to pass. And it's just a refreshment for your prior knowledge. And this is a nice video that shows the process of diffusion and osmosis. And remember that osmosis is the diffusion of water, but through a semi-permeable membrane. So for osmosis to take place, there should be a semi-permeable membrane. There should be two different concentrations, and water moves from the more water to the less water. So from the less solute to the nice video of osmosis is an excellent video. Just watch any of the two videos and write your notes and put it in your portfolio. These are the types of cellular membranes of transport across the cell membrane. I already updated the links, but if anything still doesn't work, just send me an email. I would update it again because they keep changing the links. So in this Concord Consortium animation, play the movie and leave it till the end. It will show you an actual model of the cell membrane. And then you can drag and zoom in and see more details in the model itself. Passive and active transport and the difference between them. And then finally, the rate of transport and how this helps maintaining homeostasis. Homeostasis is the maintaining everything the same. So maintaining the internal and external conditions of the cells the same, because every cell has some optimum conditions at which it can work best. So homeostasis is the maintenance of these conditions. For the Aspire to Do, it's an application of transport through the kidneys. So you already studied the kidneys last year, so it will be also a good refreshment and a good application of the knowledge that you have learned in this lesson on the kidneys. Finally, we'll have 2 quizzes. And this is your mastery check, 2 quizzes, and then the consolidation summary as usual. For the Aspire to Do experiment, if you have time, you can do it. It's for practicing some skills. And then we are going to see some applications of transport across the cell membrane, but inside structures within cells. So as we go to photosynthesis, which is the second lesson, and by the way, this is my favorite lesson, you'll find many reflections on the topic of energy, starting from here. So these are the terms that you will learn. Start with a review of your knowledge about the structure of the leaf, which you've studied in grade 11, and what energy is, just to be ready to understand what's happening to energy in photosynthesis. Engelmann's experiment, which is this video, is a nice experiment in which the color of the light that affects photosynthesis was discovered. So Engelmann allowed light moving out of the prism, which has already refracted, dispersed into or diverged into the 7 colors of the spectrum, to pass into an aquatic plant. And then bacteria accumulated around certain segments. And these segments were the segments where the highest energy of light was there, because photosynthesis took place there, producing oxygen, and those bacteria were aerobic, so they accumulated where oxygen is produced. So this way he discovered how different colors of light affect photosynthesis. And they were the red and the blue. For the steps of photosynthesis, there are 2 videos. The Amoeba Sisters video, which is the second one, is a little bit easier. Just go through them. It's a summary of photosynthesis, and if I were you, I would draw a flow chart beside it while listening to it. Finally, you have the structure of the chloroplast. It has 2 membranes around it with the stroma, or the jelly substance inside, in which the thylakoids, or those hamburger-like stacks of sacs are arranged, and these are the sacs that have in their membrane the chloroplast, and they're called thylakoids. Starting from here, you're going to zoom into photosynthesis to see more details. So you're going to have 2 activities, 2 interactives, one for the light-dependent reaction and one for the light-independent reaction. And then you're going to have in this interactive sort of wrap-up, and the quizzes are interactive as well. So for this wrap-up also, we can draw a flow chart for the 2.4.7, and then you'll have it's on Gizmo, and you will log in to Gizmo using your username and password that you have created before, using our class code, and you'll find photosynthesis. You'll do the experiment and write the lab report. Finally, the mastery check is a quiz. You're going to take to complete an interactive with questions here, and by dragging and dropping some labels, and then take a screenshot of the assignment and post it of the completed work, and post it in the assignment Dropbox. That's it. So this is your quiz, and that's it for today. I hope that you enjoy your time working on the 2 lessons today. And if you have any question, just send it to me by email. Have a great day.